So today I will talk about rare cell analysis in flow cytometry, general approaches and proper controls. And what will I talk about in detail? So we have 4 points in the agenda. First is just general things about rare cells, about the detection, what issues you have and what challenges. Then secondly, I will focus on the labeling of the cells for such experiments and the proper controls you would need. Third is then about the data analysis. So what can you do to improve your displayed data, your statistics and so on. And in the last step, I would like to go over a special method for analyzing extremely rare cells, and this is by using an enrichment. Okay, so I will start here with the first chapter. So before I talk about how to do the analysis, I just would like to define what are rare cells actually. So, and rare cells, you usually start here around 1 or 2% of frequency in your sample. And one example are dendritic cells. So 1% means, in other words, and you sometimes come across this nomenclature, means that you have one target cell in 10 to the 2 total cells. Then in human blood, you have hematopoietic stem cells. They have a frequency of 0.1% that resembles one cell in 10 to the 3 total cells. A very rare cell are, for example, endothelial progenitor cells, which is a frequency of 0.001% or one cell in 100,000. And in the immune system, antigen-specific T cells are usually found in small numbers in peripheral blood. And this is one cell in around 100,000 to 1 million cells, something in that range, resembles a frequency something between 0.001 or 0.0001. And one of the rarest cells or events in flow cytometry that you can have is when you analyze human blood and you are looking for circulating tumor cells. There you only have one cell in 10 to the 7 blood cells or total cells. And that means in 10 million, so 10 million cells, only one positive cell. And that would resemble a frequency of 0.00001%. So extremely rare. Okay, that's about what are rare cells. And then what are the requirements and issues in rare cells? So what challenges do you come across when you do it? So first, for labeling, all you want to do in rare cell analysis, you want to avoid background labeling. Yeah? Any event that is falsely positive will falsify your result in to a great degree. And for that, you would use high quality labeling reagents, so that make low background, of course, so well coupled, antibodies with a high specificity, and so on. Also in the right concentration, of course. Then you need the right controls and reagents to prevent or at least estimate the degree of background labeling. And then you might have to refine your labeling strategies to get a clear identification of the target population. So when you only have very few positive dots in your analysis graph, you have to make sure that they are really the cells you're looking for. And maybe it makes sense to not only use one marker for identification, but use a second one so that you have maybe double positive cells. Yeah, so this makes it much, much clearer than, if possible, of course. Then for the acquisition, so what is the challenge here? So of course, when you only have very few target cells and you want to see a decent population in your plot, you have to acquire a large number of total cells. So that means also your starting material, you need a decent number of cells you start with. If this is too low, you will never get enough cells to visualize. Then, of course, measuring large number of cells is time consuming. It will produce pretty large data files. Yeah? So if you have many samples, and in each sample you have many cells. This can make the analysis sometimes a little bit cumbersome. And then you always, the rarer your population is, the higher the background can become from your non-target cells, so your negative cells. So this can become an issue when you have very, very small frequencies. I will come to this point later at the last part of the presentation. Okay, so how can you shorten the measurement time? You can use a high flow rate. Yeah, just use the highest flow rate that's possible. Also make sure that the sample is sufficiently concentrated. If you have a very low cell concentration, it will take a long time to measure the sample, so maybe concentrate it as good as possible. And regarding the background, it's really important that you have a clean instrument. So it might be necessary, depending on your instrument model, that you do an extensive rinsing before you start the measurement. Then you can also do some stuff on the data analysis level to make your data more meaningful. You can exclude disturbing events. So one that you probably already know is you have to remove dead cells for a rare cell analysis. That's indispensable. So you have to do it for any rare cell experiment. And then you can remove doublets also, because doublets can also make false positive, double positive events that also can then falsify your results. And what you also can do, you can remove cells that are prone to producing background. Yeah, so what you can do is you label these. Let's say these are from your work with blood monocytes or granulocytes. So you can label them with a monocyte or granulocyte marker, and then just gate them out to avoid that these cells produce any kind of background on your target population. This is something you can also do. Okay. So a big question that you usually have, or that you ask yourself, is how many cells do I actually have to acquire to get statistically relevant results, especially in these rare cell situations where you only have few positive events. Okay, and for that I have this little graph here. On the x-axis you have the frequency of your target cells, and on the y-axis you have the number of total cells that you have to acquire in the experiment. And then you have these three lines here, and what they resemble is they represent the different statistical significant level that you want to achieve with your data. The smaller this number is, so we use here a coefficient of variation, so the less variation you want to have between samples, the smaller is the CV, and the higher it is, the more variation you get. So if you want to have an intermediate CV, so that this data has an intermediate significance or stability, then we have to follow the blue line. So how to read this graph now? So let's assume you have a frequency of target cells of 10%. That's not rare cells, that's just normal. Yeah. So then when you have the intermediate CV, you have to acquire 5 times 10 to the 3 total cells, so something between 10 to the 3 and 10 to the 4. Then, so this is what you have to acquire for that. If you have 1%, you have to go 1 decade higher, so it's 10 to the 4, between 10 to the 4 and 10 to the 5 total cells. If you have 0.1%, it's between 100,000 and 1 million cells, total cells that have to be acquired. And if you have 0.01%, you already need to acquire 5 million cells, which is already quite a lot, or very, very much actually. And this 5 million is also, depending how many samples you have to analyze later on, is also kind of the limit that you can put in a data file without having too many problems in the analysis afterwards. And if you even go below that, 0.001%, you have to acquire 50 million cells, which in my opinion is completely impossible, especially impossible to analyze. And if you need higher significance or higher trust in your data, so you need a lower CV, then you see then the values become pretty large. With 1%, you already have to acquire 1 million cells. With 0.1, you have to acquire 10 million cells. And with 0.01, it's actually already impossible. You have to acquire 10 million cells. So this is a kind of orientation that you can use on how many cells you have to acquire. and I will continue with the next chapter of today's webinar, and that is about sample labeling and proper controls. It's probably the most important part here. Okay, so when talk about labeling, what are actually the mechanisms of labeling, of background labeling and so on? And for that, I would like to go actually to talk about how can an antibody actually bind to a cell. So what are the different binding ways it does? So we have here a cell. In gray, you have here the antigens, the cell expresses on the surface. And here we have the antibody conjugates, the antibody and fluorochrome. And first of all, we have, of course, the antigen-specific binding. That's the binding we desire. Yeah, this is what we want, so no problem here. Then on blood cells or any sample where blood cells are involved, for example, if you have a vascularized tumor material and things like that, you also have the cells can express so-called Fc receptors. So Fc receptors bind the constant part of the antibody, so not the antigen-specific binding site. And in the immune system that is needed for phagocytizing antibody-opsonized pathogens. In that case, in our case, this is a technical antibody for our staining, and here it's a disadvantage that we have these Fc receptors because it will bind these antibodies, although the cell might not even express the marker you are interested in. So you get a false positive signal. Then we can have simple sticking of the antibody or the fluorochrome or both to the cell. And here, this interaction is not known, so we don't know why, which molecules are involved in this sticky interaction, so we cannot block it. So we have to see how we can handle this non-specific background, so this unknown background binding. Okay. And for that, we have different controls. So for the FCR blocking, it's quite easy. We know the interaction, so interaction of Fc part of the antibody with the receptor. So we can use an Fc receptor blocking reagent. And we have that for human or for mouse cells, and then you just incubate the cells with it, and then you have saturated the Fc receptors. Then we know another specific, another unspecific binding mechanism, and that is the binding of tandem fluorochromes to human monocytes. This is, of course, only interesting for people using human monocyte samples or where human monocytes are included. And we have a blocking reagent, the tandem signal enhancer, that blocks this interaction so that you don't get this non-specific labeling of the monocytes by the tandem fluorochromes. And this reagent contains also FCR blocking, so if you use this reagent, both things are blocked at the same time. Then, for all the interactions that we don't know, so we don't know why an antibody sticks to the cell surface, we can use control samples. So these are separate samples with the same cells. But instead of in the case of the isotype control, instead of your staining antibody, you put in a non-specific antibody, so that has no specificity for your cells, but is from the same isotype, so the same antibody class, and is coupled to exactly the same fluorochrome. And you incubate this antibody instead of the cells with this antibody instead of with the staining antibody. And then you can estimate, okay, I have so and so many positive events in this isotype control sample. So this is the background that the antibody and the fluorochrome has on this type of cells. I will show an example for this in a few minutes. Okay, then we have the isoclonic control. So you also do a separate sample with the same cells. But instead of putting in the staining antibody, you first put in an unconjugated antibody. It must be exactly the same clone, so with exactly the same specificity. And you put it into a 50-fold excess. So what you do with this antibody, if this antibody binds specifically to the target molecule, it will saturate this molecule. And then to finish the isoclonic control, after this incubation with the unconjugated antibody, you simply add the conjugated antibody, so with the fluorochrome. So when the binding is specific of the antibody to the molecules, you should not get any signal with your staining antibody, or at least a highly, if if a little bit, then maybe only just a highly diminished signal, then you know, okay, the signal I get in my normal staining sample is specific. If you still get a signal after the saturation process that you did here, that means then that the antibody sticks not onto the epitope, so by antigen-specific binding, but some other mechanism. Yeah, maybe the fluorochrome is sticky, for example. So that means that in your staining sample, the signal that you get there is probably not specific. So this is just another very good control, in my opinion, to check the background and the specificity of your staining. And this one is all of these 4 controls are very important for cell analysis. Yeah, you really need it for these kind of things that you do. Okay, so I have one example, as I promised. It's about the detection of endothelial progenitor cells. Controls that were used in this experiment is FcR blocking, yeah, because we are looking for these cells in human blood, so we have to block Fc receptor. We did dead cell exclusion. I come to that later. And we have an isotype control sample. And this here is the staining sample. Here you see we stained CD133 versus CD309. The target cells are double positive. And you see some positive cells here, very few, only 46 events, makes around 0.95% of all cells that we acquired here. So this is really truly a rare cell application here. Then I created isotype control sample that you see here. And I also stained here CD133, but I wanted to check if the CD309 staining that we see here is really specific, or how much background do I get just from the antibody conjugate. So I used an antibody that is non-specific for the cell, so isotype control. In this case, it's from mouse IgG1, so same isotype as this one, conjugated to APC. This is the same dye. And you see we get 8 background events, resembles a frequency of around 0.19%. So you see, this is then my background using the isotype control. Yeah, so we can use it for estimation of the background. Okay, so regarding the controls and the labeling, I would briefly like to introduce a new product line released by Miltenyi Biotech some time ago. And this is called the antibodies, the RIA clones. And that means all clones with this RIA name are recombinant engineered antibodies. And what are these antibodies? So they have a fully engineered protein sequence. The constant part is human IgG1. So, and this is always the same human IgG1 for all clones of this RIA product line. The Fc part, so this part here, has been mutated. So that means this antibody will not be recognized by the Fc receptors on the cells. You have one defined DNA sequence for every antibody. So it's exactly the same sequence. And when it means DNA sequence, it also means, of course, that the amino acid sequence is then the same on the protein. And it can be manufactured at highly standardized conditions, much better than normal hybridoma culture. So what are the benefits of these antibodies regarding rare cell analysis? So you have the same isotype control for all the RIA clones. So it's always this human IgG1 or this RIA isotype control. So you don't have to buy IgG2A, IgG2B, IgM, and so on. And isotype controls, you always have the same one. And the Fc receptor blocking is not necessary anymore. And this is something I will show you in a minute in a slide. So you don't need to buy these reagents. If you want to know more benefits and more facts about these antibodies, go to our website, miltenyibiotech.com/RIA, and there you will get more interesting information. And here's the example. So that shows you that you don't need any FcR blocking anymore with using these antibodies. So here I'm looking on human cells, human NK cells, CD56 positive, and we want to analyze a subpopulation of these cells that express CD158A. It's a molecule on some of the NK cells. And we use a mouse-derived monoclonal antibody, so that was the old way, or the standard way of having antibodies. Here we use it without FCR blocking, and in this example we use it with FCR blocking. With FCR blocking you have one major CD158 positive population. And if you don't use FCR blocking, you have this population as well, but you have a big additional population. So this is a really high background due to the FCR binding of the antibody, and it's really strong. Yeah? It's really a lot of cells. Now we have again a CD158 antibody in this third plot, but this time it's one of our RIA clones. So it's RIA clone 284, also conjugated to PE on the same cell sample, exactly the same cells, same donor. And here without FCR blocking. And you see, we only have this major population in this sample. We don't have this one. And we can achieve that without FCR blocking. So the benefits here for you is it's convenient, so you have no FCR blocking step, and you save money, of course, because you don't have to buy the FCR blocking reagents. Okay, so far to the RIA clones. And what other controls do you actually need? And of course, for setting up your experiment, for getting meaningful data, you always need compensation as soon as you work with more than one color, which I assume you do. And when you do compensation normally you would stain your normal cells with the antibodies you use, make single stains, set the compensation, and then you're done. When you do the same procedure with rare cells, you come into a problem, and you can easily see why. Because if we now would use in this example, that's the same example from a few slides before, we'd use the CD309 for setting compensation, so we only have very few positive cells in our blood sample. Yeah, it's only 1% in that case, or even less, yeah, so it can be even less. So it will be very hard to set the compensation here, because you have to acquire a large number of total cells, correct the compensation, acquire again to see how it looks like, and so on. So this is very cumbersome, and because these are so few events, you have a low statistical stability, and your compensation might be unreliable because of that. And so the solution is using compensation beads. So what are compensation beads? What benefits do they have? So compensation beads are kind of artificial cells. They're very easy to use. They are just in the fridge in a vial that you can simply pull out when needed. They have negative and positive beads. So negative means with low fluorescence, and then positive beads with a fluorochrome on. And these negative and positive beads have the same autofluorescence, so that's very important to set the compensation. With your cell sample, you might not know if your positive cells really have the same autofluorescence as your negative cells. They are very bright in fluorescence, so they have a very high fluorescence, so even if you have a strong expression of a marker, they usually are always brighter than your label cells, so you can always set, the compensation will always fit then. You can do lot-specific compensation if you use tandem conjugates, when you use catch compensation beads. I'll come to that in a minute. And they are useful when the amount of cell material is limited. And with rare cell analysis, you already have large cell samples. So of course, you already have to use a lot of cells. So you don't want to use it on compensation samples. And of course, as I said, very useful when you have rare cell applications, because you only have these few positive cells. Okay, so what beads are available? So we have the Max Comp bead kit 1. These are beads that are directly conjugated to fluorochrome. So on the surface of the positive beads is either FITS or PE or APC, so single stained beads. And we also have, of course, the negative beads that have no fluorochrome on. And then we have this Max Comp bead kit. And these beads, they don't have the direct fluorochrome on. They are conjugated to antibodies that are able to capture your staining antibody. For example, we have one that can capture mouse antibodies and targeting the Ig kappa chain. So this is one of the light chains. Then we have one against antibodies derived from humans, so for engineered antibodies. And we have this anti-RIA antibodies. So when you're using the RIA clones, you also have these catch beads. So you can load these beads with your antibody, and then they become positive because the antibodies carry the fluorochrome, and then you can use them for compensation. And since they also can catch will catch also the tandem fluorochrome conjugates because they're also an antibody, you can do the lot-specific compensation. So, and they're really easy to use, and you can use them on a variety type of cell samples. And that's what I would like to show you in this example. So I've used these catch Ig kappa compensation beads and loaded an FITS conjugated antibody on it. So and then I measured it in the cytometer. B1 is the FITS channel, I have the FITSy positive beads, and I plotted it versus the PE channel. In our instrument, it's the B2. It's called B2. And between the FITS and the PE channel, you usually have a pretty high spillover of fluorescence. And you can see that actually here, these are the negative beads and these are the positive beads, making, of course, a FITS signal, but also a high signal in the PE channel. So I use compensation to correct this spillover. You see the result here. So I have compensated the FITS positive cells out of the PE channel. I had to apply 7.1% of compensation. Okay, so now I used cells, leukocytes from human blood, stained them with a CD3 FITS antibody, so it's a marker for T cells, not important what kind of cells these are. And I see the same picture, yeah, so I have spillover of the FITSy positive cells into the PE channel. And now I use the compensation, so the 7.1% applied to this sample, and I get this picture. So cells are nicely compensated. And now I have a different cell sample, a cell line that is stained with an epithelial cell marker. It's a cell line with epithelial marker on the cell surface. And you see here, these cell line, which is often with tumor cell lines, the negative cells already make a very high fluorescence in this channel, but also in this channel here. And the positive cells are here. And even with these cells that have a completely different autofluorescent properties, my compensation derived from the beads will fit because I now apply this compensation and get this result. And you see they are nicely compensated. So these compensation beads can really be used for a variety of different cell samples. Okay, so the next part I would like to talk about is, so what can you do on the data analysis level to improve your data for rare cell application? And one is that probably many of you know already is the exclusion of dead cells. I recommend to do it every time you work with living cells, but it's indispensable when you use rare cells. So dead cells generate false positive events because they bind any kind of reagent non-specifically. You can stain them with a dead cell dye to identify these dead cells. In the example I show you, I use propydium iodide, but there are many different dyes possible and available on the market, like DAPI, fixable live dead stains, zombie stain, and so on. There are different names for many different types of dead cell stains. Okay, so but we stay with propydium iodide. It normally binds to nucleic acids. But when you have a living cell like this one, the entry into the cell is blocked or highly diminished. And the cell stays negative, so it's not labeled with this fluorochrome. If the cell is compromised, so if the cell is dead, the membrane is open and the dye can enter this cell and can bind to these nucleic acids. So, and then in the plot you see, these are the unstained cells, so these are my living cells, and in the diagonal we have the dead cells. And what you can do now during your gating strategy, you can do a so-called dead cell exclusion by drawing a region around the living cells and go further with these cells in the analysis. And because since you included only the living cells, the dead cells, they disappear from your analysis in your later analysis steps. So when you really look onto the specific markers. Okay, and as I said, For rare cells, as analysis, dead cell exclusion is indispensable. When you don't do it, you cannot trust the data at all. Yeah, so it's really, really important to do it. Okay, then another issue that can occur in cell analysis is doublet formation. So when cells are simply sticking together. In flow cytometry, it's normally a single cell analysis, or is designed to be a single cell analysis. So doublets can really falsify your results, and especially if you have only very few target cells, so like rare cells, you can get big errors in your data analysis later on. I have one example here that's not specifically a rare cell analysis, but nicely shows how what the effect of doublet can be in data analysis. So we have here a stem cell culture, human-induced pluripotent stem cells. They were cultivated with mouse feeder cells, so it's a xenoculture, so it's different species. And you need these feeder cells to nurture these iPS cells. We harvest the cells by dissociation from the tissue, and then label the cells, the iPS cells, with TRA160, that's an antibody for these pluripotent cells. And the murine feeder cells, we use a mouse-specific antibody that stains a marker on these cells. And you see here, this is the stem cell population, TRA160, and this here is the feeder cell population. And then you have quite a number of double positive cells. And actually, it's a little bit odd, because we stained here with an antibody against the human antigen and with an antibody against the mouse antigen, so you should never get double positive single cells. But these aren't single cells, these are doublets consisting of a human and a mouse cells, and that's why you get this double positive signal. So how can we get rid of these doublets? And for that, you can exploit an electronic feature of the cytometers. So you can plot, for example, the side scatter parameter on one axis of the dot plot using, or the density plot using, side scatter area versus side scatter height. So height is a different aspect of your measured signal. You can also use width if you don't want to use height. So but one must be area and one must be height or width. What you then get is a diagonal of cells. These are your singlets, and these singlets, why are they in the diagonal? Because when they get a higher granularity in the height level, it also grows proportionally in the same way in the area level. But doublets have a proportionally higher area value in the side scatter than in the height parameter. So they are not in this diagonal anymore, and you can see them here as cells coming out of the diagonal. So these are now the doublets, and when you make now a region around the cells in the diagonal, you include the singlets, but you exclude the doublets. And now we plot the singlets, so in this region, again onto our fluorochrome staining. So same channels here, TRA160 versus feeder cell marker. And now you see most of these doublets that we have here in the ungated, in the non-doublet excluded example, they are now disappearing here and are removed. We still have some doublets, because it might be that the difference in granularity for a certain doublet is not big enough to fall out of this diagonal. What you can do is you can also plot them after this step. You can plot them again also on the forward scatter height versus forward scatter area, and then make an even more restricted singlet analysis. And then you will see that some of these doublets probably also disappear. So you can do that on even two parameters, one after another, and then you get the most precise picture. Okay, then we come to the last part of the presentation, and that deals with the analysis of ultra-rare cells. So if cells are really, really rare, for example, like these CTC, these circulating tumor cells, and these cells can be only visualized by enrichment. Why do we need this enrichment? Why is the visualization so problematic? We just would have to acquire more cells. That would be the logical compromise. The lower the frequency, the more you have to acquire cells. And this is shown on this slide. So here we have a histogram with two cell populations, negative cells, positive cells. So this is a normal high frequency, let's say it's 50%. So you have two equally proportioned populations of histogram peaks. When you have now a rare cell assay, especially with very rare cells, so think of these CTCs or these endothelial progenitor cells that I mentioned, here you see you have a very, very large proportion of negative cells, so of your non-target cells, because you have to acquire a lot to see your positive cells. And then you have here your rare cells, and now you see what the problem is. The background of this negative cell peak grows the more cells you acquire. So it gets wider and wider, and at some point it partially or completely obscures your rare cell population. And now it's not possible for you to set the gate really correctly, because if you set it here, you include some of your negative cells. If you set it somewhere here, you lose too many of your population. So you can't really do gating. And this occurs quite often if you have cells below a frequency of 0.1%. So 0.1% is roughly the detection limit of a cytometer. And below that, this problem becomes more and more drastic. Yeah? So then it will become more and more difficult to see the cells. Okay, so we can visualize these cells much better if we do an enrichment. What enrichment methods are available? So you can do selective expansion in culture. This is the most unattractive method in my opinion. So first of all, it's extremely time consuming. You have to wait for days or even weeks to expand your target cells. And what is more striking in my opinion is that they will definitely change their phenotype and function, yeah, because if you cultivate them in vitro, they will change definitely. And you don't have the ex vivo picture that you might want to see. Then you can use centrifugation techniques, only possible for some types of cells. Endothelial cells, for example, is sometimes possible to use centrifugation techniques, but usually you lose a lot of cells during centrifugation. Yeah, 30-50% is no problem to lose with a density gradient. And of course, if the sample is not very big, then you can also lose too many cells. Yeah, then you don't have enough cells left to analyze in the end. So then you have flow cytometry sorting. It's time consuming. Yeah, so you have to probably start with a quite large cell sample because you're looking for very rare cells. So you have a decent starting population. So this can take hours or almost a day to get the cells sorted. You need an expensive equipment. Yeah, flow sorters are easily several €100,000 expensive or even more. The instrumentation is complex. So most people need an operator. It's not so easy to operate a cell sorter. And you still have the background problem. Yeah? So it's really hard to set the gate that you really get a clear population at the end. So what we recommend for this purpose is immunomagnetic enrichment. It's easy to perform. Everyone can do it. You don't need an operator for that. And you can do it very quickly. It's independent of the cell number. Yeah? It doesn't matter if you separate 1 times 10 to the 8 or 1 times to the 9 or 1 times 10 to the 10 cells. It always takes the same time. So it's bulk enrichment. It's not single cell enrichment like flow sorting. And it's gentle to cells. Yeah, so you can imagine if you sort for hours in a flow sorter, this is a little bit more stressful, also with the high pressure and so on. So this is a very gentle way to do it. Some of you might already know our technology, our Max microbead technology that we use for magnetic enrichment. I just go over it briefly to illustrate how this technology works. So you have 3 modules. The first module is an antibody conjugated to a magnetic particle. The second one is a magnet, and the third one is a column. So the process that you do is you label your cell suspension here. You label your target cells that you want to pull out with this conjugate, with this magnetic particle conjugate. Then put it onto the column that is located in the magnet. The column will retain the cells. and the magnetic cells. And when you take the column out of the magnet and flush it out, you get your magnetic cells out. In that case, this would be maybe your rare cell population. And a few words to the components. So the microbeads are very small. They are 50 nanometer only in diameter. That's around 200 times smaller than a cell. And because of this very small size, there is no risk of activation by receptor cross-linking that would happen with larger beads. So even if you do a positive selection, so if you really target your rare cells, you don't get any influence on the cells. Because of their small size and they are non-fluorescent and so on, they are fully compatible with downstream applications without the need to remove the beads. So you don't have to remove the beads for that. And for example, you can use them right away in flow cytometry, for example, for this pre-enrichment, or you can use it in culture for molecular analysis. They don't disturb. They're super paramagnetic, so when they're out of the magnetic field, they're not magnetic anymore, so you can easily flush the cells out of the column. They are biocompatible and non-toxic, and they are even used because of all these features in humans, so in clinical research to develop cell-based therapies in humans. One word for the column. So the columns contain the steel ball matrix. You can see that here. These are very small steel spheres. Between the steel spheres, the cells are held in fluid suspension, and the space between the spheres is really large, so the cells can pass this lumen between the spheres without being squeezed through the column. Yeah? So there is no pressure onto the cells. What is the function of these columns? So they amplify the magnetic field by a factor of 10,000-fold. And this makes it possible to even retain these very small beads. Otherwise, if you don't have this amplification, you would need a pretty large magnet. And furthermore, since you have this very strong magnetic field inside the column, you only need minimal labeling of the cell. So we don't have to cover the cell with this antibody magnetic particle conjugates. It's not necessary to do that. Usually, depending on the expression level, we only cover 20 to 30% of the epitopes on the cell. Yeah, so you don't need to coat the cells actually with these particles. It's very flexible. You can have it for different cell numbers, so smaller columns for smaller number of cells, larger columns for higher number of cells. You can use it in basic research, but also in clinical applications, as I mentioned. And you get with this column excellent purity, recovery, and viability. Okay, and what would be the workflow now for a rare cell analysis experiment using this pre-enrichment? So you start labeling your cells with the microbeads and maybe already with your fluorochrome conjugates. Then you put them onto the column, your sample. You enrich the rare cells here on the column, and then you elute the rare cells and subject it to the measurement on the cytometer. And then you get a rare cell population. So how does you get your rare cell analysis? Quite easily. So how does that look like on the data? And I'll come back to these histograms I've shown before. So in the original sample, rare cell sample, you have here your negative cell bead and your target cell peak. And you see here, this is the situation I've shown you. Part of the rare cells are obscured by the negative cells here, by the background of the negative cells. So what you do is you label the target cells, you pull them out by immunomagnetic enrichment, just as I showed, and then you elute this positive fraction and measure it in your cytometer, and then you get a picture like this. And now you have a decent frequency of these positive cells, and the negative population peak is now highly diminished. So it's much, much smaller, less background, doesn't obscure your positive population. And now you can analyze this data much more easily than without enrichment. I have one example, that's the detection of circulating tumor cells in human blood. And for this experiment, we used a cell line, a tumor cell line, the same one I showed with the compensation. These cells express CD326, and we spiked it into different numbers of these cells into 10 million white blood cells. So we didn't, here this is a sample without spiking, with 10 cells and with 100 cells. Then we labeled the sample with CD45 via blue, so CD45 for the leukocytes, and CD326, a fluorochrome conjugate for the tumor cells, and also with microbeads, so magnetic particles for this. So then, without enrichment, we just measured the fluorochrome labeled sample, and we see we don't see any cells, even when we spiked in 100 tumor cells. They're not visible without enrichment, so we get 0 positive cells. So there are no tumor cells detectable because they are so few. So you have only 100 or 10 cells into 100 million total cells. They are not visible, no matter how many cells you acquire. Then we use the magnetically labeled samples and enrich them onto the column. And that is shown in the lower row. And here you see the number of CD45 positive cells is diminished, so there are not that many cells anymore in the sample. Where we haven't spiked in cells, you don't see any cells. Where we have spiked in potentially 10 cells, we get out almost 10 cells, one more. So it's a 10% error maybe. And where we spiked into 100 cells, we get 89 cells out. So we get almost all of the cells out that we spiked in. So here we have around an error of 10%. This error can also occur during the spiking, because when you just spike in 10 cells, you can easily make a small pipetting mistake. So that's actually quite challenging to do it in the right way. So that shows how reliable this technology is, that you really get more or less the same number of cells out that you had before in the original spiked sample. And you see here different cells, number of cells have been spiked in, and you see that you usually recover almost the same number of cells that you spiked in before. So yeah, it's nice to see that this works quite well. Okay, so then another example I would like to show you, and this time a natural example, so not a spiking experiment. So when you're working in immunology, for example, vaccine development and so on, you're often interested in the frequency and in antigen-specific T cells, and there is the problem that the frequency of these cells is really low. So here are some different populations of antigen-specific T cells. For example, naive antigen-specific T cells, so that have not participated in any immune response so far. You have something around 5 times 10 to the 4, so 1 cell in 5 times 10 to the 4 blood cells, down to 1 cell in 10 to the 7. So they're really, really rare and really, really difficult to visualize. For example, cells against an allergen, can be slightly more frequent. So if a chronic allergy can be something around 5 times 10, 1 cell in 5 times 10 to the 3 blood cells, down to 1 cell in 1 million blood cells. Here for vaccine development can be a little bit higher depending when you boost it. 1 cell in 100 down to 1 cell in 10 to the 5. And for a virus, it depends very much on the virus, chronic, acute infection, and so on. It can be really frequent, like 1 cell in 50 maybe, down to 1 cell in 1 million. So it's very hard to analyze these antigen-specific T cells because of these low frequencies and this wide variety of frequencies. How can you analyze antigen-specific T cells? Just briefly how they work for those of you who are not familiar with T cells. So T cells have a T cell receptor that can recognize a certain epitope of a pathogen. So each T cell can only recognize one epitope. This epitope is usually presented via an antigen presenting cells that has degraded the pathogen into small peptides. And this peptide epitope can then be recognized by the T cell. When this happens, the T cell secretes cytokines, they express activation markers, and they proliferate and so on. And there are 2 different ways how you can analyze them. One is the analysis doing multimers. So what you do is you produce artificial MHC molecules, so the same molecule you have on the antigen-presenting cell. You load it with the peptide. Usually you couple it to this MHC chemically, to this MHC molecule, and you make multimers, so several, here this is a tetramer in that case, several MHC molecules, and they are labeled with a fluorochrome. So this multimer binds to the T cell receptors of the cells, and then you can visualize the T cells. This is one way to do it. It's a good way to do it, but it's also expensive and it's not very flexible, because for each peptide you want to analyze, you have to produce a multimer, and the production is takes a lot of work, and therefore these multimers, when you buy them, are really expensive. Usually one vial for a few tests is 1,000 euro or even more. The response that the T cells do when you stimulate them, you can stimulate them simply by loading the different types of peptides or the antigens onto the APC, and then the T cells respond with cytokines, so you can measure the cytokine response, or you can measure them according to activation markers, and that's something I would like to show. So on CD4 T cells, when they are activated, they express the molecule CD154, or it's also called CD40 ligand. And this, with using this molecule, we can analyze these cells, and we can also enrich them, because only the activated cells express this molecule. And that's what's shown here. So what we did is we stimulated blood cells using a lysate from Aspergillus. That's a common mold, that's, for example, also problematic with allergies. And then we enriched the Aspergillus reactive CD4 T cells, the antigen-specific CD4 T cells, by using CD154 microbeads, so against these activation markers. And this will only enrich these Aspergillus reactive cells. Then we stained here CD154 versus CD4, so T helper cell markers, or a subpopulation of the T cells. And here on the top row, we have no stimulation of the T cells. And you see, without stimulation and without enrichment, we only get a background labeling of 0.02% of cells. So that's actually nothing. It's clearly below the detection limit, so it's really nothing. If you do an enrichment, we get 2% of positive cells. That's when you enrich the background simply. You only have a very weak positive cells here, on very few only. Okay. Then here we stimulated now the cell sample, and this is the original sample, so without enrichment. So we already have almost 20-fold of frequency of reactive T cells, and you already see the expression is also much higher than what you see here. So you have brighter cells. And when you enrich, you really get a very decent high frequency of cells. It's 20% of these cells expressing CD154. And these are now the Aspergillus reactive antigen-specific T cells. And now you can do even further more stuff with these cells. You not only can detect them, but you can also analyze their cytokine expression, for example. So again, we have here CD154 versus CD4. It's a different experiment, but the same procedure, also the Aspergillus lysate. We have here enrichment of 29%, so 30% from almost nothing. And now we stained here again CD154, and this time versus a cytokine, so intracellular cytokine staining. This is here interferon gamma per CpSI 5.5, and you see we have a frequency of 4.2% of interferon gamma positive antigen reactive T cells on the enriched sample. If we, without enrichment, we were only able to detect 0.05%, so a very, very low frequency, already also below the detection limit. So here the statistics might not be very robust in that case. Here we stained IL-10, another cytokine involved in some regulatory processes sometimes. And here we see on the enriched cells, it's only frequency of 0.6%, but we have a more or less clear population. But without enrichment, it was only 0.007%, and that is really something you cannot visualize with really decent statistical significance. So here, the enrichment, without enrichment, you would not be able to analyze these cytokines on the T cells. Okay. You can automate this enrichment. So for that, I would briefly like to introduce our cytometer, the Max Quant instrument, to you. So it has many, many different automated features, so measuring from 96 well plates, automated labeling, and so on. That's not so important for now. I would like to go on two features that have to do with the rare cell analysis, and that's the thorough intersample washing. So the uptake needle and the measurement path is washed extensively after each sample automatically. So by this we have a very low carryover from cells from one sample to another that is below than 0.01%. So that means you don't get any cells from the previous sample. Very important for rare cell, yeah? So you don't have any contamination with false positive events from another sample. And it contains an enrichment column so that you can enrich microbeads labeled cells. Yeah, so they can be loaded onto the column. And the convenient thing is that after the enrichment, the instrument will automatically elute the cells from the column and automatically will perform the measurement, the flow cytometric measurement of the cells after the enrichment. So the workflow has become now much, much easier. So you label your sample. Then you give it to the Max Quant. The Max Quant will automatically take it up, automatically enrich it, and automatically analyze it in the flow cell, in the actually flow cytometer part of the instrument. So you just put in your sample, press start, and you can walk away, and the instrument does everything for you. And I have one example from such automated enrichment. These are circulating endothelial cells from human blood. They are characterized by the expression of CD34, which is actually a hematopoietic stem cell marker. So they are together with the hematopoietic stem cells in the CD34 positive population. They express an endothelial cell marker, that's CD146, and they do not express the leukocyte marker CD45, so they are CD45 negative. So we use these three markers to identify these cells. So we label the cell sample with CD34 microbeads to enrich them later on in the process, so to get an enrichment of the circulating endothelial cells. And the controls that we use is FCR blocking, isotype control. Then from the data that I show here, we excluded the debris by using on the scatter, and we did a dead cell stain, so we excluded the dead cells, so gated on the living cells. And we stained in the top row CD34 versus CD45. So you see here, these are all the leukocytes. And here you have the CD34 positive cells. This is the original sample, so without any enrichment, just the fluorescent labeling. And we have here a frequency of 0.03% of the CD34 population. So already a rare cell, clearly. And within this CD34 population, so within this region here on the top, we have these endothelial progenitor cells that are displayed here, and they should show up here in this region. So CD146 positive, CD45 negative, so in the upper left. And you see without enrichment, you get like 2 or 3 dots. So not statistically reliable. Now we do an enrichment, and instead of 0.03%, we have 21% of cells. And when we plot these enriched cells onto our CD146 plot, we really see a decent number of these CECs and can analyze them now. And I have made an isotype controlled sample, enriched this as well. You see there are CD45, CD34 negative cells, but I made here an isotype control for the CD146, and I can see that there's a little bit of background, but not that much. So I can estimate the background for this rare cell application. Okay, so last slide for today. When you're doing rare cell analysis, maybe you want to try the enrichment method and you have questions for that, you have issues with the enrichment, you can contact our sales force. We have a very well-educated sales force that can help you with technical hints and technical counseling. We have also instrument specialists if you use our instruments that can help you to set up the application. We have a dedicated technical support and instrument service that you can contact. And for our Max Quant instrument, we have a 24-hour online support. So a remote support that you can contact even at nighttime, so when you have to work at night, which I don't hope happens too often. And we have a Max Academy Training Center in Germany and in the US that offer face-to-face training courses. For example, for this T-cell enrichment, we have a course where we're doing this T-cell enrichment using the CD154. And you will find the offer training courses here when you follow this link. So methylnebiotech.com/maxacademy.